going to be focusing on movement in the cell and movement of a cell from place to place. And so the majority of the lecture will focus on the cytoskeleton and motor proteins. At the very end, we'll talk about the extracellular matrix. Before we get to that, however, I have to introduce your first homework, which is going to be available right after class today and will be due on Monday morning. And this homework is in the activity type that we call Learn Before Lecture, so it's LBL 1. And we started instituting these a couple of years ago when we had problems that the students were consistently getting wrong on the exams. And every year we'd say, okay, they just don't get this concept. And in lecture, we'd change the lecture around, and we still wouldn't be able to get that concept across. And so we decided we needed more time in class and we needed to do different things. And so the strategy we're using here is we're offloading some of the material I would have just told you on lecture slides in class, so you have to learn it before class. This basic information then you're going to learn in this particular case, it's about the nucleus and ribosomes. And it is going to not just be a reading assignment, but it's a worksheet to help you go through that reading and look at the figures in the same way I went through the reading and looked at the figures to prepare the 4 lecture slides I used to have at the beginning of lecture number 5. Those slides are no longer there. You learn the material in the Learn Before Lecture, and we take that time to do additional things, and we're going to have a bunch of things that we do in class that will hopefully help you learn that activity, that concept, at the depth that we want you to understand. Okay, it should take you about 20 minutes to complete. Today after class, you're going to download an LBL worksheet from the website. It is a Word document. Then you can type your answers into that Word document. But to upload it, you need to convert it into a PDF. You can work with your friends on these. That's fine, you can talk to each other, but you have to do your own work and you have to upload your own LBL. If you upload your friends, then you won't get credit for the assignment, and that is considered academic dishonesty. So you can work together, but you can't just copy their answers. Okay, they're due Monday morning at 9 a.m. You're going to upload them. This is on your EEE. You'll have a Dropbox that will say Assignment Dropbox, and there will be instructions about how to upload it. Complete it before you do Lecture 4 quiz, because on Lecture 4 quiz, there'll be 4 questions from this lecture, plus 2 questions about the material that will be covered on the LBL. So you'll have to have done that before you take the Lecture 4 quiz. Okay, and don't wait till the last minute when people are trying to do it at the last minute and the computer crashes and then you uploaded it at 9:10. You don't get credit for it. So do it well ahead of time. Okay, now we're going to get started on the content for today, which is the cytoskeleton. And this is a network of fibers that extends throughout the cytoplasm of all eukaryotic cells. And it's important for 2 major things: structural support in the cell and motility. So what I mean by structural support is it's important for helping to maintain the cell shape, as well as anchoring internal components within the cell. So often in the book and in many of my diagrams, there will be a cell and there will be a nucleus and things scattered throughout the cell, and they kind of look like they're floating free in the cytoplasm. In fact, that's very rarely true. For the most part, things are at the right place in the cell, and they're directed and kept in that place by interaction with cytoskeletal elements. Okay, motility, what I mean by that is movement of a cell from place to place, like our white blood cell that was chasing after the bacterium, or the bacterium that was running away, or movement within a cell. So in last lecture, we talked about a couple of proteins, like an aquaporin channel. What does that do? What's that for? Movement of water across the membrane, that's right. So it's in the plasma membrane. Is it made in the plasma membrane? No, it's made in the Raphiar. We'll explore that in more detail in the next lecture. So it's made in the Raphiar inside the cell, but it works out in the plasma membrane. So it has to get transported to the plasma membrane, and the cytoskeleton is really important in actually getting things from one place to another in the cell. Okay, so there are 3 major classes of cytoskeletal fibers. actin, microtubules, and intermediate filaments. In this class, we'll focus only on actin and microtubules. When you get to cell biology, you'll add in and think more about intermediate filaments as well. Okay, actin and microtubules are very different molecules. They're made up of different components, but they have some really important similarities. And I'm going to go over their similarities first. And there are 4 of them that I need you to know about. The first is that they're both composed of multiple protein subunits. And these assemble to form fibers. So these are proteins because they're composed of protein subunits. That's an important thing to remember. And in the case of actin filaments, these assemble. We have an actin protein subunit illustrated by this one small green ball here. And they assemble together to form a 7 nanometer filament, and it sort of looks like a string of pearls that's twisted together. Microtubules are composed of tubulin subunits that are assembled together. So tubulin and actin are both proteins. They're assembled together, and we get a microtubule here, and it's a hollow tube because of the way these tubulin subunits assemble. Okay. They are also both dynamic structures in that they undergo rapid assembly and disassembly inside the cell, and this is happening all the time in the cell. Each of the fibers has ends that have specific and unique properties. Here we have actin, and actin has a plus end and a minus end. These pluses and minuses do not denote charge. This is a bit confusing, and we will have plenty of times that you'll see pluses and minuses in this class that are associated with charge. In the case of cytoskeletal elements, this denotes where subunits are added and subtracted. In the case of actin, subunits are added at the plus end, and they're removed at the minus end. So the fiber grows by adding actin subunits to the plus end, and it shrinks by removing them from the minus end. In contrast, and something you just have to memorize, is in microtubules, they also have a minus end and a plus end. But in microtubules, the minus end is most often anchored in a particular region, here in the microtubule organizing center, the MTOC. And the majority of addition and subtraction of the tubulin subunits to make this grow and shrink occurs at the plus end. So actin is growing by adding at the plus end, removing at the minus end. Here we're growing by adding at the plus end, and we're shrinking by subtracting at the plus end as well. Okay, and the 4th thing that they have in common is that they both interact with motor proteins to generate cell movement. Myosin is the motor protein associated with actin, and kinesin and dynein are the motor proteins associated with microtubules. Okay, so I'm going to show you this movie just very briefly again. We have a white blood cell moving around. And when we talked about this before, right, we recognized that this region, the tail region of white blood cell, is not a flagella, but it's the uropod. And there are changes in cytoskeletal elements in this moving cell that are important for generating the movement. Okay, so what we're going to do now is think about where the cytoskeletal elements are organized in the cell and use this to help make a prediction about which of these cytoskeletal elements is important in the movement of the white blood cell and the extension of the pseudopodia during phagocytosis. So here is a cell where I've labeled the actin fibers in red and the microtubules in blue, and you can see they all lay on top of each other. It's a bit hard to see where they are. But if you separate them, you can see that the actin has a very high density around the cortex of the cell, and then sort of a meshwork in the rest of the cell, whereas microtubules have a sort of a radial array, where here's the nucleus, and the microtubules are radiating out toward the plasma membrane. So the question is, which of these, the actin or microtubules, are involved in the extension of the pseudopodia and the process of phagocytosis? And you can discuss this. You're going to have to explain why you chose which one you chose. Okay, click in if you haven't. The majority of you have already done so. Okay, very interesting. So it's evenly split between microtubules and actin. Okay, and microtubules was a little bit more, I think. And so a group that said microtubules, give me the reasoning why you said microtubules based on what you saw there, the movement of the white blood cell. Yes, and say it really loud.
Because you can, the microtubules can go pull in and they can pull back out with the membrane. That's why I chose it, 'cause it's more radial. So
Okay, good. So you said, I see the microtubules are radial, and those pseudopodia are definitely sticking out, right? And so they could potentially be sticking out and being at the center of those pseudopodia. Yeah, there's another. Microtubules connect with motor proteins. Yes, that's absolutely true as well. Okay, how about a group that said actin? Anybody want to volunteer why you said actin? Yes.
we thought actin was more concentrated on the outside, so it's better for phagocytosis.
Okay, so they picked location. Actin is on the outside, so they may be closer to those pseudopodia and be able to be involved in that process. So that's a good choice or logic. Yes, one more. Which one, the actin? Okay, so that you're saying, well, the actin is assembling by adding at the plus end and subtracting at the minus end, so that might be more associated with this movement. Okay, that's a good logic. And so what I'm trying to do is, you don't have to know the answer. I haven't told you the answer to this, but use the knowledge that you have. How about, think about something that you know from biology and actin and myosin from high school. What is that involved with? Muscle contraction. What is that? Is that a type of movement? Yes. So that's another piece of evidence to suggest that maybe it's actin. And in fact, actin is the correct answer. But all the people that said microtubules and used the logic behind where they saw what it was doing, that's what you're supposed to be doing in this class, is using that information to actually try and make a prediction. Okay, so now actin, so what happens actually is, right, there is a, I'll show you in just a second, but there's interaction between actin and myosin that's important, and as well as assembly of actin that somebody else brought up. Okay, we're going to start with actin and its interaction with the motor protein myosin in the context of muscle cells, because this is something that many of you are already familiar with. And so we're going to start here, and then we're going to move on to movement of a white blood cell, that type of movement. Okay, actin and myosin organization is shown below. So here's a muscle cell, and we're going to look at one unit of the muscle cell here, which is designated between these two Z discs, this one here and this one here. This region is a mirror image of this region. So if we look at this region, we have actin that has a plus end and a minus end. The plus end is attached to the Z disc, and the minus end is in the center of this unit. On this side, the plus end is attached to the Z disc, and the minus end is here. So this is a mirror image of that. In between, organized by these purpley blue structures, those are the myosin motor proteins. And that's not a single myosin motor protein, that's a bundle of a whole bunch of myosin motor proteins. Okay, so what happens is, in the presence of calcium, these heads of these motor proteins will bind to actin, they will exert force, and they will pull in this direction on this side, and in that direction on the other side. So if I was standing right in here, and I'm the tail region of the motor proteins on this side, our myosin heads are on this side, and then we have myosin heads on this side. In the presence of calcium, they bind to the actin, they exert a ratcheting force. That means what space decreases? Right? That space decreases because these Z discs move in. Okay? And then and these are plus end directed motors, so the myosin grabs toward the plus end and pulls in. Okay, so that causes a shortening of the muscle. And we call these plus end directed motors because the myosin heads are always moving toward the plus end of the myosin. Myosin heads are moving toward the plus end of the actin. I'm sorry. Okay. Now we're going to look at just one of these little, let me go back and show you. We're going to look just at this region right here so you get a little bit better picture of what's going on. Okay, we have our actin filament, plus end is attached to the Z disc. Right below it is another actin filament, plus end attached to the Z disc. And this is the myosin bundle. And here is a single myosin molecule that has two globular heads and a twisted tail region. And these heads are what bind to the actin filament. Okay, so just a quick question. Myosin binds to actin either when ATP is present, myosin binds to actin and moves toward the minus end, myosin binds to actin when calcium is present. Go ahead and click in. All right, I'm going to close the window. Okay, very good. 65% of you are correct when calcium is present. So to illustrate this process a little bit more clearly, we developed a demonstration a couple of years ago, and I'm going to do it again. We actually have some new twists this year, and so I'm going to need two TAs are going to come down and be our Z discs, or Z lines, and I need 4 students to come down and be myosins. And if you volunteer for this, remember it's going to be videotaped, so you're going to have to sign a release, and if you have a skirt or a dress on, you probably don't want to do this because you have to sit on the floor. So, anybody over here want to do this? Yes, I'll have one person on the end and one person in the back next to the person in the green shirt. Yes. Yeah. You. Yeah. Okay, and then I want 2 from this region in the back. Anybody want to do it? Yes, somebody in the very back there, and somebody in the very back on the end right there. Great. Okay, come on down. So we are going to have 2, and your name is Stephen, very good. Okay, you are going to be a myosin. We're going to get you set up. And why don't you sit right here on this and face that direction. Good. And your name is? Ashley. Can you come down and be a myosin on this side? And you're going to sit down and put your feet right up next to his because they are forming this region.
I know I already volunteered, but you could choose someone else if you want. But
Pardon?
I know I already volunteered once.
Okay, so does anybody else want to do it? Okay, yes, you're right there. Okay. Okay. And your name is? Vincent. Why don't you sit down right behind her with your legs crossed? That's okay. It's right up next to her. Okay, and I need my Z lines to come up and get sort of in place. And you are Rebecca, stand right behind him. Okay, so now what we've created down here, just sit with your legs crossed and right, you can sit, right behind him. Okay, good. And actually, cross your legs, you need to be a little bit close, like Indian style. Okay, okay, and I know I need to be back here so you guys can see. Okay, so now we need to orient our Z lines, and so we have Rebecca over here, and she needs to have her plus end at the right place. Where is her plus end going to have to be? In her hand, right? Okay, and that's up above. Yes, very good. And she's going to actually go this right about there. Good. Okay, and then Lindsay is going to have her plus end right about there. Okay. Okay, so here's our active element with a plus end. Okay, so now they're not bound. Why are they not bound? No calcium, exactly. They are going to each need to have two globular heads. What do you think are going to play their globular heads? Their hands, exactly. Okay, so I magically sprinkle calcium in, and what happens is there were revealed yellow binding sites, which are the binding sites. Her yellow binding sites are revealed when I added calcium. They turned. And now the globular heads can go up in both hands and can bind onto a yellow binding site. Bind onto a yellow binding site, all of you globular heads. Yeah? Okay. Bring it down for her. Okay, very good. Okay, not only do you bind, but you ratchet. Not really far, but a little. And what move? Okay, so actually, let's start back a little bit like this. Okay, now, a little bit further back. Okay, a little bit further back here. Let go. It's okay. Okay, now. Okay, so now bind. Okay, now, when you bind, you not only bind, but you ratchet a little bit. So go forward a little bit. And your Z's come in. No, stop. Okay, stop. Okay. Now, in fact, how that we need to have you let go, and the only way you can let go is you have to take some ATP chocolate. Oh, very good. You need some too. Okay, good. And once you eat that, which you don't have to do right away, then you regenerate, and now you can move toward the plus end of your fiber. So the plus end is that way, so grab a binding site that's closer to the plus end. Yes? No? Good. And now ratchet forward. Good. Okay. And then ATP. So this sort of occurs at random, the ATP. And then you regenerate and ratchet, and that's how you decrease the distance here. What is this distance here called? The H-band. Okay. So now all the calcium is gone, and what happens? You relax. So let's do this one more time. Go back further. Okay, so let's see. And so now we're going to, we'll just, okay, so now there's no calcium in, so there's no binding sites. We sprinkle on calcium, we get activation, we get exposure to the binding sites, globular heads go up and ratchet a little bit, okay? And then some of them, in the presence of ATP, let go, regenerate and ratchet again, and pull, okay? And you regenerate and ratchet again. You always move your hands toward, and then you close that H-band. Okay? Makes sense? Okay, calcium's gone. Let go. Very good, and you relax back. Okay, very good. Oh yeah, you have to sign releases, and so do you, TAs as well. Okay, let's move forward. Okay, we've already done this. Myosin binds to actin. What, A, B, or C? C, when calcium binds. And this is something that people often forget, that it's not when ATP. You have to have ATP to get this cycle to go, but in the presence of calcium, the myosin binds and ratchets, and then only when ATP comes in does the myosin unbind from the actin and the cycle goes again. Okay. Not only is actin and myosin interaction important for this muscle contraction, it's also important for cell shape in non-motile cells. So we have a three-dimensional dense actin mesh, which is what you sort of saw in that picture that I showed of the organization of actin. And it's just inside the plasma membrane, and so it gives some structure to cells. And we also have in motile cells, actin and myosin being important in the crawling or amoeboid movements that you see. So amoeboid movement, it can be an amoeba that's moving, and it can also be a cell that's moving like a white blood cell. Okay, so how does that work? Here we have a cell in which this is the leading edge and this is the trailing edge. And at the leading edge, for this movement to occur, the first thing that needs to happen is disassembly of the actin mesh at the leading edge. So at the minus end of the actin filaments, you're going to have disassembly. And so up in this region, it's not such a tight mesh, it becomes loosened. Actin and myosin then interact very much the same way they do in a muscle cell. You have actin filaments, myosin motors that bind to actin filaments and exert force and cause a contraction. And that happens at the trailing edge. And when that does, what happens when you squeeze basically at the back of a water balloon? It goes out the front end, right? It pushes toward the front end. So you're squeezing things up here, and also you're detaching this region. And then actin filaments are actually pushed up into this area, and they assemble into the developing pseudopods, and they actually form sort of like the cytoskeletal elements or the structural elements of the pseudopods. As they are pushed out forward, then there are actin filaments that are assembled in there. Okay, so that's termed amoeboid movement, and now looking at this and understanding these cellular elements and these interactions, now you know much more about how that white blood cell was moving around and chasing the bacteria. Okay, we also now need to move to microtubules and the interactions they have with their motor proteins, which are kinesins and dyneins. Microtubules are also important in cell structure and shape. And what we're going to be talking about most here is their role in providing a track for movement of components within the cell over long distances. So if this is a cell and the plasma membrane, then we have a nucleus in here. Microtubules in a non-dividing cell are oriented in a very particular fashion in that the minus ends are near the microtubule organizing center, and the plus ends are out toward the plasma membrane. The movement along these involves these motor proteins called kinesin and dynein. Okay. While I set up a second demonstration, I want you guys as groups to identify critical features of a motor protein that would be necessary to move this green vesicle in my stick cell here to the plasma membrane. Whenever I do this exercise, students say, well, what do you mean? What kinds of things are you looking for? And I'm really looking for very simple things like this motor protein is going to have to be able to move, to actually move this vesicle from where it is to the plasma membrane. The motor protein is going to have to move. What other features does this motor protein have to have? So think about that. I'm going to ask you about while we set up the demonstration, and then we'll go over it. need to go here. So first, what other things does this motor protein have to be able to do to move this vesicle from where it is to where it needs to go? Yeah.
It has to be able to bind to the vesicle.
Very good. So it has to have a binding site in which it recognizes the vesicle. So there's something on the motor protein that has to recognize the vesicle. What else? Yes. It is going to need energy because it's going to have to move. So it's going to need energy. That's true. Yeah, and you had?
It has to be able to bind to the microtubule.
Exactly. So it has to have two binding sites. One is the binding site for the vesicle, and one is the binding site for the microtubule. Really good. Okay, and what else? What's one other thing? We have to get it from here to here. Yes?
A protein has to know which one's the plus side and which one's the minus side.
Exactly. So there has to be directional movement. It has to be moving in an oriented fashion. So in fact, we do have motor proteins, the two motor proteins, kinesin and dynein, are directional proteins, and kinesin moves only to the plus ends of microtubules, and dynein moves toward the minus ends of microtubules. Okay, so I need a volunteer to be dynein, way up in the back in the middle. You just raise your hand in the white cap. And somebody right in the middle here too. Yeah, you have a orange picture. Good. Okay. Oops. Messing up my cell. Okay, well they're coming down. I have my cell. It has some aquaporin channels in its membrane. What is this, do you think? Nucleus surrounded by endoplasmic reticulum. What's this then? The Golgi. And what are these green things? Microtubules. Okay, very good. And your name is? Carmen? Carmen's going to be kinesin. And so, and your role is going to come up in a little bit. And Carmen is going to need to insert a spiky protein that's inside a pink vesicle into the plasma membrane. Okay, so she's going to have to be in the cell as a motor protein. And she has to find her pink vesicle. She's going to have to bind to it, so she has a binding site on her hand, right? And then she has binding sites on her feet that can walk in what direction? Plus end on a microtubule. You can choose any microtubule. But you really need something else. What does she need to move? Energy, which she gets from chocolate in the mitochondria. Okay? And now she can move. And kinesin walks along microtubules, and now she's gotten to where she needs to go. But now we're going to insert by the process of what? How do we get the spiky protein into the membrane? Exocytosis, exactly. So you're going to exocytose it, right? And now it's on the plasma membrane. Okay, very good. So you can put that down there. Okay, so now kinesin, oh, stay there. Because she can't really walk backwards, right? She can only walk on plus ends, and all the minus ends are there, and all the plus ends are here. So she and all her friends, all her kinesin friends, would, if they all only walked in the plus end, then what, toward the plus end, and they all inserted membrane, what would happen to the cell over time? It would get bigger and bigger and bigger, and sometimes you want a cell to get bigger, so that can happen. But in fact, if you want to maintain a cell of a certain size, then you have to have also what process? Taking membrane out of the cell. And endocytosis, right? And so we need to have, your name is? Tony is dynein, and Tony can actually do the process of endocytosis. He has to be inside the cell to do this. Okay? And he is going to do endocytosis with this cell, with this vesicle. So, how do you do endocytosis? Show your classmates. Ah, very good. Now you formed a vesicle. Okay, and you need a bit of energy as well, which gets from the mitochondrion, right? It's chocolate from the mitochondrion. And now you can walk toward the minus end along microtubules. Very good. And you know, really interesting, you know, with the way you're walking, that these guys actually move in a fashion that is literally, that's sort of how they move like this and like that. And so it's not a complete straight line. It's not just a directed line. But now there's also movement that if you all ended up here, if dyneins are all here, kinesins all here, then you'd be stuck. But in fact, there's movement of dyneins and kinesins that you would be moving on vesicles that have lots of dyneins and moving in that direction, and you could be moving on vesicles that have kinesins moving in that direction. So you can get distributed throughout the cell. You don't all end up at one place or another. Okay, very good. Thank you very much. Okay. Oh, sorry, you need to sign up. And if the TAs could come and help put this away, just fold it up, that would be great. Okay, while we go forward, because we have more stuff to cover. Okay, our microtubule motors are kinesin. You can just leave them where they just leave everything in there. Just fold it up. Where kinesin is moving toward the plus end, dynein toward the minus end, and they're moving vesicles and organelles, so mitochondria are being moved back and forth, vesicles, also chromosomes during cell division. Important point though is kinesin's always moving toward the plus end, dynein always toward the minus end. Okay, microtubules and dynein are also important in movement mediated by cilia and flagella. And the organization of these structures is really important. So the microtubule doublets run parallel to the length of the appendage. So if this was the flagella or cilia, then what happens is these are organized in this parallel array, and we have the minus ends, which are anchored in a region called the basal body, that is the attachment between this appendage, the cilia or the flagella, and the plasma membrane. It's a similar organization to the microtubule organizing center, but it's at the plasma membrane, not inside the cell near the Golgi. Okay. Also, we have these doublets are linked by two proteins. We have these blue linking proteins, which are shown here, and we have dynein motors. Shown here, the heads are on this side and the tails on that side. Which direction does dynein move?
Negative.
Toward the minus end. That's right, the minus end is anchored in the basal body, and so when they walk toward the minus end, and these are still linked together, they're not allowed to slide because of these blue linking proteins. What happens? They bend. Because as the motor proteins are walking down and they're still linked together, then the appendage bends. And that is what mediates the movement of the flagellum that propels the bacterium as it's running away from the white blood cell. What happens when you have one of these moving in a cell that's fixed in place, like a cell in the lining of your digestive system or in your respiratory system? What's it doing? It's moving fluid across the surface of that tissue or that cell. So in a fixed tissue, the cell moves fluid over the tissue. Okay, we also have the extracellular matrix, which is present in the extracellular surface, so right outside the plasma membrane of all animal cells. And it also plays a really important role in structural support of the cell. So along with actin and microtubules, it's important in cell shape, structure of the cell, but it's also important in transmitting signals from the outside to the inside. So I'm not going to say much about this, but the major components of the extracellular matrix are glycoproteins. And we've seen glycoproteins one other place, right, that were actually in the membrane. These are glycoproteins that were made inside the cell, but they were excreted. So now they're on the outside of the cell, and they have they're proteins that have lipids on them, sugars rather, proteins that have sugars on them. And so the major glycoproteins we have are collagen, indicated by this big blue or purple structure, and that is actually one of the major structural support fibers that's found in the extracellular matrix. We also have fibronectin, which is shown here, and these are kind of interesting because these are connecting the extracellular matrix to a transmembrane protein called an integrin. And you can see this transmembrane protein is actually attached to actin inside the cell. So movement out here and a signal out here can get transmitted into the cell by the interaction of fibronectin with integrins that are in the cell membrane and that are interacting with intracellular actin filaments. Okay, so for the last 5 minutes, I want you to do this exercise that is similar to what we've been doing on Piazza, and it's to take an old exam question and answer it where you give a true or false for each of the possible answers and explain in 1 sentence why. I want you to do this as a group, and then you're going to turn in your cards at the end. So, and as always, if you have questions, just raise your hand and I'll come up and answer. Okay. We're running out of time, so if you could pass your